Welcome back to everybody. The break was very short, but we needed to start again because all the speakers needed to have the planned time. So I rapidly introduce Chiara Dall'Asta. Chiara Dall'Asta is a chemist and she had a PhD in food science. She's a professor at University of Parma. And so I give the floor to Chiara that can add something if she wants. Please.
Thank you. Thank you very much, Paola, and welcome back, everyone. So I'm really pleased to be here today as well and to present some part of what we work on over the past year. So I have the task today, which is a hard task, to introduce to the role of plants in chemical mixture. Let's say if we can just a brief outline of what we will see over the presentation. I will try to explain why biotransformation of plants is so important or must be considered in risk assessment, or at least in the science at the back of risk assessment. I will use modified mycotoxins as a case study, so to explain in a concrete way what plants may do to chemicals. And of course, I will discuss with you the impact in terms of risk assessment or exposure. And I will discuss with you about the challenges, opportunities that plant biotransformation may offer to our field, so to the risk assessment, but in particular to food science and what we can expect or may expect for the future. So, what we know about co-occurrence. I think that Bruno and Armando and Jean-Loup already told you everything. If we consider mycotoxins, but this can be said for any kind of chemicals, but if we consider mycotoxins, we can say that we have many mycotoxins that may be produced by the same pathogen, by the same fungus. We can have different fungi on the same crop, but we can even have different mycotoxins, but we can even have different chemicals, different residues, dioxins, pesticides, or whatever. And we have many crops in our meals. And if we have a pasta with ragu, as we have here in the slides, you may have contaminants, environmental residues, mycotoxins, but also veterinary drugs coming from the meat. So, of course, co-occurrence is a reality of our everyday exposure to chemicals. Anyhow, this picture, this narrative is lacking. And the relevant actor that we still miss is the plant. And when I talk about the plant, I talk about the crop. So what plants, edible plants, may play as actors, or how they may act towards multiple chemicals. This is what we address today. So, the plant. Plants are living organisms, and any kind of living organism, plants are able to uptake nutrients, but even contaminants. Pay attention to the words of Jean-Loup just before the break. If we consider the exposome, we have to consider the contaminants, but even the nutrients. So all the chemicals we are exposed to. The same for the plant. The plant are living organisms. They absorb nutrients from the soil, from water, and of course from the environment. They bring these chemicals, then we will talk about contaminants, these chemicals within their cells, and they of course use these chemicals for doing a large spectrum of processes, biochemical processes. What we call contaminants or residues in the context, in the frame of risk assessment, are just xenobiotics for the plants. And xenobiotics are well known to be uptaken according to their polarity from soil, from water, translocated, so moved, for example, from roots to the leaves, from the leaves to the other edible or other tissues in the plants, and then metabolized. Let's check what metabolism in plants looks like. Of course, we don't have a liver. You know, liver is the organ for metabolism in mammals, but in the plant we don't have a liver, but we can consider a kind of green liver system because the plant is doing pretty much the same to chemical as other more common or more familiar mammals, animals are doing. So plants may uptake chemicals from the environment, soil, water, atmosphere, bring into the cell, then start to exert phase 1 and phase 2 metabolism, so pretty much hydroxylation and conjugation, with the aim of getting a more polar compound. Why? They cannot excrete this more polar compound through urine, feces, or breath like we do, but they can use the vacuole and sequestrate or just put the chemicals into the vacuole. The vacuole is a kind of trash bin for the plant. So they just uptake the chemicals, transform them, and put them into the trash bin, into the vacuole. Usually, the phase 1, phase 2 metabolism in plant has exactly the same goal of our metabolism. So it's a way to decrease toxicity. And in terms of plant, we call it phytotoxicity. So it's the toxicity, the toxicological effect of the chemical toward the plant. But the point is, and you have to keep in mind very clearly, is that what is less toxic for the plant may not be less toxic for mammals or humans. So we are not the same organism. Even insects, don't forget the insects. So plant scientists know the metabolism of plants since decades, dozens of years. And the ability of plants to modify xenobiotics was studied a lot in terms of enzymatic machinery, in particular for resistance towards pesticides. These two plants that you have in the slides, the black grass and the ryegrass, were the first documented cases of metabolic resistant, CYP-mediated, so again, P450 mediated reactions towards herbicides. So these plants were able to resist the action of planticides due to a very efficient metabolism. So it was known by the plant scientists. It was known also by the environmental scientists. The ability of plants to uptake contaminants from soil and biotransform them is called phytoremediation. It's a strategy. It's a very, very important green strategy to clean polluted soil, polluted waters. You know, you can put plants or, plants mainly, into polluted water to get it cleansed again. For example, from heavy metals. The point is that when it comes to edible plants, so when plants are edible plants, so our veggie, our vegetables, the phenomenon can be of concern. Just for instance, the phenomenon or well-known phenomenon of cadmium enrichment in rice is pretty much the same. The rice is able to capture, to uptake the cadmium from the soil and fix it into the, bring it into the plant, the edible tissue. So what is called phytoremediation from an environmental science point of view is a masking from a food science point of view, from a risk assessment point of view. Because when it comes to edible plants, all the chemicals in the environment can be uptaken, biotransformed, and in this way, they can escape the routine analysis. This is an important point. You discuss about occurrence, but if you don't have the analytical data, if you are not able to detect analytically the compounds because they are changed in terms of chemical structure, then of course you lose a part of the bigger picture in terms of exposure, in terms of contaminants. So this is known, as I told you, is known from an environmental science point of view, is known from a plant science point of view, but is less known from a food science point of view. This is a study from last year, first studies in the field of food science, let's say, about the ability of edible plants, in particular vegetables, to uptake a pharmaceutical drug, Diclofenac. It's for your back pain. from the water and bring it into the edible tissue. In that case, the scientists found more than 30 metabolites of diclofenac in salads, tomato, carrots, onions, and whatever. So, I mean, maybe in terms of amount, concentration, these 30 metabolites are not, how can I say, not relevant in terms of exposure and risk assessment, so contribution. But from a scientific point of view, we cannot neglect them. We have to consider them. This is something we have to keep in mind, at least at the back of our mind. Okay, so not only diclofenac, but very recently, other reports on antiretroviral and antiviral, pharmaceutical drugs, nanoparticles, sunscreen ingredients in edible plants, so cosmetics. Still, all papers and data published in a context of environmental science. So again, we miss the food perception of this problem. And here is the reason. Unfortunately, we have to say that scientists talk a lot within their community, but discussion and exchange between different fields, different communities, is often difficult. In particular, it was more difficult in the past year because of not availability of open science and whatever. So you have to consider that knowledge coming from one field does not easily enter other fields. And this is exactly the story of modified mycotoxins. Back in the 80s, you know what mycotoxins are. Armando already gave you a flavor of the story, so I don't want to really spend too much word on that. But in the 80s, David Miller, which is really a prominent scientist in our field, reported an interesting information. He was following the accumulation of deoxynivalenol in wheat in the field. He was following the accumulation during the growing season, and what he saw was that deoxynivalenol concentration reached a peak and then decreases. So he said, it was 1983, he said, I observed a fast decrease in DON after a peak in naturally infected compounds. Maybe this strange or unexpected trend is due to the breakdown of deoxynivalenol driven by plant enzymes. So somehow he hypothesized a metabolism, but it was not proven. The technology, the methodologies, the tools available at that time didn't allow him or the community to go on this road. After 20 years, can you understand? The first report appears in the scientific community. It was about DON3-glucoside. DON3-glucoside is the first masked mycotoxin, so metabolite, plant metabolite of a mycotoxin, identified and isolated so far. It was the first. It was deoxynivalenol, metabolized by the plant, stored in the vacuole, and, let's say, invisible for analytical methods. From that moment on, the road was really long. And nowadays, we don't talk about, or we don't refer to this kind of compounds as masked mycotoxins, but better as modified mycotoxins. So there was a paper in the 90s with scant data, then the milestone paper in the 2005, then again another milestone paper in 2012, till the EFSA opinion in 2014. In that opinion, there was an umbrella concept written, which is extremely important. So the definition of modified mycotoxins. Modified mycotoxins are those mycotoxins that are structurally, chemically modified as an effect of a metabolic process exerted by a living organism, plants, but also other fungi, but also mammals, or as an effect of food processing. Masked mycotoxins are in sensu stricto, so really in a really focused way, narrow way, those compounds obtained by plants, so metabolized by plants. So mass mycotoxins are included in the definition of modified mycotoxins. For the first time, EFSA realized or stated that modified mycotoxins were important, not only, but EFSA also stated that risk assessment should take into consideration also this class of compounds because they may more or less contribute to the overall toxicity, so to the overall toxic load of your food. So we need to consider them. What is known now in 2021, after 7 years from the first EFSA opinion? There were other opinions in between. We know now quite clearly that pretty much every mycotoxin, every parent mycotoxin, so mycotoxin produced by fungi, once entered the plant may be biotransformed. So from one mycotoxin, you may obtain a pool of modified mycotoxins. And the spectrum of these modified forms are really different according to the genetic background of the plant. We also know that these compounds are not cleaved in the stomach of mammals. They are less toxic per se, but they can be totally cleaved in the gut of mammals by the resident microbiota. So they can really contribute at least for a little part, or more or less little part, to the overall toxicity. And finally, we know that the biotransformation occurring in the plant may be affected by the biotic and abiotic stressors, so by the environment. So different climate condition, different soil condition, different genetic background, different bacteria on the plant, other fungi, other chemicals may affect the metabolism. So this spectrum of modified mycotoxins may change. And this is pretty much the same for any other metabolite of chemicals formed in the plant. Is it a challenge for us in terms of risk assessment, food safety, food science, or is it even an opportunity? Plants modified mycotoxins to get rid of them. This is extremely important. As a part of their natural defense system. This is wonderful for breeders because their biotransformation in plant minimizes the spread of the fungus. Therefore, breeders nowadays may use this ability to biotransform to select or to breed more resistant or resilient crops. This is definitely good in terms of food security and resilience and sustainability of our crops, but and the overall mycotoxin load in this way is reduced. But we have to take into consideration that in this way, the proportion of mass mycotoxins towards, let's say, parent mycotoxins becomes more prominent. So we need to have analytical methods and we need to consider these compounds in our risk assessment. Otherwise, we miss a part of the picture. Even fungi, which are important actors, may adapt to the environment. So they may adapt themselves and their biosynthetical processes to the new plants, to the new bred plants. So we have already some reports about fungi that have changed the spectrum of mycotoxins in a way to produce mycotoxins less prone to biotransformation, to be biotransformed by the plant. So, you know, it's a war, and there are two living organisms that are interacting, and we are right in between, and we have to understand what is going on. It's definitely difficult. And last but not least, little to nothing is known so far about the modulation of plant biosynthetic pathway machineries by the Cooccurrence of different chemicals in soils and water. So we are getting more polluted. Climate change is changing, is becoming heavier and heavier. So what is going on? What is going on in the plant, and how the spectrum of different modification will change in the future? This is extremely important, not in terms of current risk assessment, but in terms of a perspective risk assessment. So what we need to be able to do in 5, 10, 15, 20 years. Coming back to the starting picture, what we know about Cooccurrence. You know already the story, but I hope now you know that we missed something. We missed all the chemicals, we missed the environment, we missed how the plant may interact with the environment and with mycotoxins and fungi as well. So we need to adopt a system approach. There are already cases of system approach in this field. I don't want to go into this, but we have a project granted by the Parma Foundation, and there's a lot of things going on in this project, but one is extremely interesting. We are trying to use new perennial grains, which are plants, wheat, with a long root, and the root stay in the soil for years. So it's not sealed every years, sown every years, but it's just the roots stay longer. They are really long and they stay longer in the soil. So this is a more resilient crop. This is able to get nutrients from the soil, but from another point of view, the roots stay in the soil longer. So what is going on with these kind of plants when they face, I don't know, pollution, flooding, extreme weather events, chemicals in the environment? We need to consider all the faces of the coins. There are not two. Sometimes it's not a coin, it's a dart. So just to wrap up, the assessment of multiple chemicals in our diet is much more complex than expected. It was already complex before the break. I hope I was able to add another layer of complexity. Fundamental science is needed. This is definitely fundamental science. This cannot be brought to risk assessment immediately, but we need to understand the mechanism to tackle the complexity and to be ready, to be prepared. Proper risk assessment methodologies should be implemented under the One Health approach. And when I talk about One Health, I always consider plans. And what is definitely more important to me, we need to move from One Health approach to One Science approach. So we need to talk definitely more within the bigger, the whole scientific community. I think this school is an attempt to bring different point of view to you as attendees. So economic, food science, plant pathology, risk assessor, regulatory frame, everything. This is something we need to push and really support for the future. So I hope I was able to give you the overview in the time I had, and I hope not to be too long. Okay, thank you.
Thank you very much, Chiara. You used a bit more time than you had, so I limit my comment just to underline the concept of One Health, which is quite important in this context, and to stress the high resilience of plants and fungi that can make the framework where we are working on much more complex.